immense pleasure to have Dr. Jack Gallant here for our Brain Awareness Week. Jack Gallant is a professor of psychology at the University of California at Berkeley and is affiliated with the graduate programs in bioengineering, biophysics, neuroscience, and division science. He received his PhD from Yale University and did postdoctoral work at California Institute of Technology and Washington University Medical School. His research program focuses on quantitative computational modeling of the brain. He is known for his discovery of the curvature tuning in visual area V4, for pioneering the study of single neuron responses during natural vision and attention, and for his work on quantitative, computational, and statistical modeling of nervous systems. Over the last 10 years, he has developed a new computational modeling approach that is optimized for human functional neuroimaging data. The recovered models accurately describe how the human brain represents information during natural tasks, and they can be used to decode information in the brain in order to reconstruct mental experiences. His brain decoding algorithms were named as one of Time Magazine's 2011 Best Inventions of the Year, and he appears frequently on radio and television. So without further ado, I'll hand it over to Dr. Jack Gallant.
Thanks, thanks very much for inviting me. I want to tell you, for professors, the biggest honor for us is getting invited somewhere by the students. Because professors invite other professors somewhere all the time, and who really cares? The people we're here for are you, and it's really great to be invited, especially by the undergrad students. Because lots of times I tend to think I'm just talking in an echo chamber. It's nice to know that at least once in a while somebody else is paying attention. I'm going to be talking about MRI today, and in particular I'm going to be talking about one particular study of speech comprehension. This is a really small group, and I'm a pretty accessible guy. So if you have a question, just either raise your hand, or if I don't see you, because I'm old and my eyes aren't working so well anymore, just scream, and it's fine. I'll just stop. We'll just deal with it then. Okay? You don't have to wait till the end. You might as well deal with any questions you have right away. So let's start with a little background of what the problem is. This is the structure we're trying to understand. This is a big, meaty computer. I'm a computational neuroscientist, and I can think of this like a big computer. It's just made of meat stuff rather than electrical stuff. And our job is to reverse engineer this computer. Now, the brain's kind of weird because it's not like a normal computer. In a normal computer, you have hardware and you have software, and they're kind of independent. In the brain, you have a bunch of hardware, and the wires within about 20 minutes or half an hour are fixed. So all of your thought occurs on a fixed set of hardware. And as you're sitting here listening to me and your thoughts are changing over time, the hardware is fixed. So that must mean that somehow, or rather the subcomponents of the hardware are interacting to give rise to different kinds of emergent thought. Right? And there's a bunch of parts of the brain. There's this folded sheet over here called the cerebral cortex. This is just like an outside shell that's folded together and sucked into a very small volume. And that's just so your mother can give birth to you. You want your head to be as small as possible, right? Then there's this very old structure, the cerebellum, that is involved in motor coordination, timing, and other things. We'll just ignore that. And then underneath here in the middle are a bunch of subcortical nuclei, like the thalamus and the pulvinar and other things. These are very old brain structures, and I refer to these as the giblets, and I just kind of ignore them. So for me, I pay attention mostly here to the cerebral cortex, the outside of the brain. And this is the part of the brain that has expanded hugely in mammals. What happened is, somewhere or other in evolution, a few million years ago, evolution figured out, hey, wait a minute, if I make this modular architecture and this cortical sheet, I can just make it bigger and bigger and bigger and keep adding stuff to it and just make it as big as I want and very efficiently make a much more sophisticated kind of computer. And this is kind of a change from the old way evolution used to work in more primitive animals, where every time you wanted to make a new brain area, you had to sport off a new subcortical nucleus with its own complete architecture and wiring. So this cortical architecture scheme is a very neat evolutionary innovation that has appeared and been elaborated mostly in mammals, us and our primate cousins and rodents and things like that. Okay, so if you look at the architecture of the brain, there's a few technical limitations. We're cutting off the bottom, say 1/8 of the display. It's usually not important, and I'll explain what's going on when you can't see it. Just try to ignore it. The brain is organized at multiple scales. There are single neurons. These neurons are organized in your cerebral cortex into a 6-layered sheet. This is also a columnally organized sheet. If you look down at the surface of cortex, say from the outside of the brain, looking down on the brain surface, you'll see that the brain actually has maps. This is actually an orientation map. The different colors here refer to neurons that have different orientations. These aren't single neurons. This is kind of a low resolution picture. And if you zoom in just on this little square here, and you do calcium imaging and look at orientation tuning, you can see that the individual neurons, they suppose are shown here, this is work from Clay Reed, are tuned for orientation. And this particular map of orientation is in primary visual cortex, the largest single area of the brain and the first cortical stage processing for visual information. Now there's more than just one map in the brain. There are, people guess, based on scaling issues from our lower animal cousins, probably about 500 different brain areas. Right now we know about 100 or so of them, and we know these from anatomy. So what we hear is an old picture some of you may recognize from the beginning of the century from Brodmann. This is old Brodmann's maps of the brain areas as determined only using light microscopy. So we took humans after they died, sliced up their brain, we looked through a light microscope and he tried to see where the neural architecture changed across cortex. And he came up with around 100, 110 different brain areas. But the true number of brain areas is probably 5 times this large, we expect, just based on a variety of animal studies. Now, when I said there are maps, the thing to remember is every part of the brain, the cortex, that we know about, there is always a map. There are tons of parts of the cortex, like all of prefrontal cortex. We know very little about it. We don't really know exactly how it's organized, and there are no known maps in those areas. But all the areas that we say, I feel like I have a clue what's going on in this part of the brain, there's always a map. So this is an orientation map in primary visual cortex, where color is showing you different orientations. There's also an ocular dominance map, which is left and right eye. There's a spatial frequency map. I think this is a temporal frequency map. All overlaying each other, all sort of superimposed on this two-dimensional sheet. So a neuron in primary visual cortex is tuned for many, many different parameters: orientation, wavelength, spatial frequency, temporal frequency, x, y position. You add all these up, you get like 15 parameters that the neurons are tuned for, but they all have to be represented across this cortical sheet in this two-dimensional map. So when you take a high-dimensional space and project onto a low-dimensional space, you get these fractured kinds of maps. Right? Here's a tonotopic map in primary auditory cortex of, as I recall, a mouse. Here's a whisker map, also in rodents. Rodents have whisker barrels, individual fields for individual whiskers. I have lots of whiskers. I do not have whiskered barrels because this is a rodent-specific structure. Yes?
So when you say map, I assume you mean that the color is related to increased activity in this area? Is that what you mean?
By a map, I just mean that some dimension, some parameter, is represented in a spatial distribution across the cortex.
Okay.
So as you move across the cortical sheet, you get different values of that parameter. Yeah. So for vision, we know kind of what these parameters are, right? For primary visual cortex, the primary organizing dimension is spatial position on your retina, because there's a retinotopic map in primary visual cortex. But at each retinotopic location, right? Because your eye's like a camera, right? So the image comes in, it's projected on the back of your eye. The projection on the back of your eye is pointillistic because the photoreceptors are just point samplers. That information goes down your optic nerve and then is projected essentially onto the surface of the cortical sheet. But instead of being represented just in points, at each location on the cortical sheet, you're actually representing all the orientations and different spatial frequencies and a lot of other information that is conferred by aggregating information across the individual photoreceptors in the retina. Right? So the point is, as far as we know, all of cortex's maps, they're just maps of different kinds of information. So there are a lot of visual maps, there are a lot of auditory maps, there are a lot of motor maps. For vision, we actually have a pretty good, there we go. For vision, we have a pretty good idea of what the maps are for primates. This is primate work done in the 1970s, 1980s, and early 90s by a variety of anatomists. Here on the upper right, we have the brain of a macaque. This is a side view of the macaque's brain. This is a medial view, and they've colored in the cortical visual areas here. And then the macaque's brain is, of course, folded up inside the skull, which is very inconvenient. So anatomists like to flatten the brain. You can imagine if the brain is like the the brain's like a beach ball that's had all the air sucked out of it. You can stick an air hose in there, inflate it into kind of a brain-shaped beach ball, and then put some slices in it and flatten it out. If you do that to a macaque, you'll end up with something about the size of a large cookie. If we do this to your brain, we'll end up with something about the size of a large pizza. And the different areas are like, tiling this pizza, right? So you can see the different areas in the macaque here. They're colored in different visual areas. And you can see that the macaque, about half of its brain is visual. When this slide was made, there were 30 brain areas that were known for vision. Now there's probably about 40, 45. These are actually hooked together in a complicated network. Here's a wiring diagram of the macaque visual system. This was known in 92. Here's primary visual cortex here, visual area 2, visual area 4, and this complicated network. The top of the tree in this particular diagram is the hippocampus, which is in the middle of the brain, but there are a whole bunch of networks for different brain subsystems. There's going to be a network for the motor system, for the auditory system, for prefrontal cortex, which is involved in abstract thought and planning, and so on. These areas are organized hierarchically and in parallel. You can see that the wiring is very, very dense. And each time there's a wire that goes from one brain area to another, there's also a feedback wire. So this is a really complicated, highly connected network. In the macaque monkey, it's known that there are about 383 areas total that we know of so far. This is getting near the end for the macaque mapping. It's probably going to end up being, there are going to be about 400 brain areas total. And each one of these brain areas has a different job. It represents a different kind of information. It plays a different role in mental computation. Scaling issues suggest that in humans, the number of brain areas is about 500, but we don't know where these areas are. So if we take the human brain, which is inconveniently folded up, we put some slices in it and we flatten it out here. Here the dark areas are what's in the sulci, the folds of the brain, and the light areas are what's in the gyri, the part that you can see of the cerebral cortex. And the problem we have here is, okay, where are the areas? We want to know where are the areas, and we want to know what they do, right? If the macaque monkey has 40 visual areas, we probably have like 60 or 70 visual areas. Because unlike a macaque, we can actually talk about the things we see. So we probably have a bunch of visual areas that are involved in us being able to name things and talk about them, right? We can think about the future, macaques think about the future too. They live in a matrilineal hierarchical society, and they're very smart about their location in the pecking order. So they're very good at like social cognition, but they don't think too much about, say, building jet engines or things like that. That's a human kind of thing. So humans probably have a bunch of other like long-range planning areas and parts of the prefrontal cortex. You can see why humans probably have a lot more brain areas than the macaque. Our brain is 10 times as big, and we have a lot more hardware. So the problem that I have in my lab is, where are the areas here, and what do the areas represent? What information is represented? I'll explain quantitatively what I mean by representation in a minute, but really you can think of this as the pizza problem. I essentially have this pizza structure, and the areas are like the pepperonis on a pizza, but I'm just looking in the pizza in the dark. I don't actually see where the pepperonis are, and I have to have some scientific method to go in and find all the pepperonis in the pizza and figure out what they do. So until recently, until 20 years ago, 1993 to be exact, we had no way to really deal with this issue with humans. We did have some brain imaging technologies like electroencephalography, which you've seen these kind of hair nets that people wear that kind of record electrical activity in the skull, in the brain, excuse me. But those methods of measuring brain activity are really bad for a variety of reasons that I'm happy to go into if you want to know why, but it will hopefully become clear in a second. But in 1993, it was discovered that this tool that we had for measuring anatomy is actually a tool developed for chemistry originally in World War II, and then by the 1970s or 80s, it had been modified to be used to look internally inside people's bodies to look at anatomy. It was discovered in 1993 that you could use this tool to measure changes in blood flow, blood oxygen use, and blood volume that occur as a consequence of brain activity. And actually, so in MRI, actually, you can measure two different things. So you can measure the structural connections between brain areas. This is using a method called diffusion imaging. Diffusion imaging, it uses an MRI machine to measure the flow of water molecules, essentially protons, right? Stuff associated with water, flowing through the brain. And remember, neurons in the brain are just a sheath of fat with water and seawater in the middle. And water wants to flow along a neuron, along a dendrite or an axon, because that's just a water channel, but it doesn't want to flow across the membrane because that's fat. So when you have these fiber tracks, one area projecting to another, the water tends to flow along that fiber track and not across it. And you can use a special MRI method to find the direction of water flow, and then you can infer the locations of brain connections from these data. Now, you may have seen these pictures before. This is a great technology. It's really the technology of the future. I would argue that this is not the technology of today. And the reason is, although this method makes really beautiful pictures, there's actually no way to know how accurate these pictures are. There's no way currently to put error bars on your diffusion imaging pictures. So you get a picture, you can show people, hey, look, I got a picture. But if they ask you, well, how good is this picture? You go, I don't know, pretty. So it's kind of bad. It'll get there, but we're not there yet. Right? People are working on that problem. There's another method you can do, which is called functional MRI, and that's shown over here on the right. In functional MRI, we're not trying to reconstruct the pathways. What we're trying to do is take the cortex and divide it up into little tiny volumetric pixels called voxels. And in each voxel, we measure the change in blood flow, blood oxygen, and blood volume, and that's hopefully somehow related to brain activity. So MRI is really a miracle. fMRI is a miracle technology, but it has some problems. The first problem is we're measuring blood flow, not neurons. So here's a little diagram of what it measures. Here's a presynaptic cell and a postsynaptic cell. And we're going to have some neurotransmitter that's bound up in these little balls called vesicles. Vesicles bind with the presynaptic membrane. They release neurotransmitter. It floats across the synaptic cleft, and it binds to the postsynaptic membrane. Now, you don't want to just leave extra neurotransmitter. Once all these binding sites are full, you don't want to leave extra neurotransmitter in the middle here just floating around, because it'll keep reactivating the postsynaptic cell, and you'll become paralyzed. That's actually how nerve gas works. It's very bad. So you have to get rid of this extra neurotransmitter. You have to clear it from the synaptic cleft. So evolution, being very ingenious, came up with this kind of Rube Goldberg vacuum cleaner system, where it has astrocytes that surround the synaptic cleft. They actually bind up the extra neurotransmitter. They re-deliver it back to the presynaptic cell so it doesn't go to waste, it's recycling. And then they have little feet that go around the blood vessel, these little philopodia. And when the astrocyte detects that some neurotransmitter bound to the receptors, it releases its philopodia. This makes the blood vessel bigger, and this means that more blood can flow through the blood vessel. Why is that? It's because neurons are engines, and they're actually chemical engines that run by a chemical called ATP. And to make ATP, you need to extract sugar from the bloodstream, and you need to burn that sugar with oxygen to make ATP. So this is essentially an anticipatory mechanism. The astrocytes say, okay, I detected some neurotransmitter. Somebody around here just fired. That means somebody's going to need more ATP. So I'm going to release my philopodia and allow more blood flow to go through to provide the raw material for these neurons to make their fuel. Pretty ingenious. But the thing to remember about this is we're not measuring neurons. We're not even measuring anything close to neurons. We're measuring something really complicated, non-linearly coupled to neurons. And so instead of thinking about the brain and recording from the brain, you should think about this. This isn't a Brillo pad. It's actually an extraction and a plasticized map of the blood supply to the brain. And this is actually what we're measuring here, this blood supply. Now, it turns out because of the way MRI physics works that the resolution we have to do this is fairly low. For those of you who are in neurobiology, remember that as far as anybody knows, this cortical sheet is made up of these little tiny columns that span the 6 layers. The columns are about 0.5 millimeter, 3/4 millimeter across, and there's hundreds and hundreds of thousands of columns in this kind of columnar structure that makes the cortical sheet. And since we're measuring from a blood vessel about the size of a voxel, which is where most of our signal is going to come from, we're actually going to measure a lot of these cortical columns. So this is a little model of the visual cortex called the ice cube model. Some of you who have taken vision saw this model. These are ocular dominance columns. These are orientation columns. These little white things are color blobs. And around the color blob, there might be 6 or 8 columns in this area. And we're going to record from something about this size, a hypercolumn, which as you can tell is going to be like, 10, 15 cortical columns. So we're going to lose all the local information that's being encoded in this structure. We're going to get the more kind of low-scale information. And the big future push to MRI is to get it down to a resolution where we can measure a single column, something about, say, 600 or 700 microns across. When we can do that, we can actually use this as a neurophysiology tool. The problem is just when you breathe and your heart beats, your brain's moving, say, a millimeter inside your skull all the time. And that movement means if you're trying to record a given location, 700 microns, you've got this thing moving in there all the time, it's difficult. So it's technically hard. And finally, this thing is really slow. So a neuron can fire about 100 times a second. And this map here shows the blood flow response, which in MRI is called the BOLD response, for blood oxygen level dependent response. This is the BOLD response to various locations in the visual field that are stimulated in this checkerboard pattern. And these colors here indicate which locations in the visual field cause a significant change in the BOLD response. You see the blue thing here makes the BOLD go down. All these other places make the BOLD go up. But if you notice the time here, it's about 4 seconds after the stimulus. So a neuron can fire 100 times a second. A BOLD signal takes 4 seconds to occur. That means you're losing a huge amount of information, temporal information. Right? So if you kind of do a calculation, an optimistic calculation about how much information we're covering in MRI, it's about 1/1,000,000th of the information available in the brain at any given point in time. And it's probably actually worse than that. So you might think, why the hell are you wasting your time with this? This looks like it sucks. Why should we waste our time with a method that sucks? But actually, MRI has one good thing about it, and that's that MRI measures things in space, in 3 spatial dimensions. So EEG, electroencephalography, measures things at the surface. It's measuring a 2-dimensional signal. And to figure out where those signals came from in 3D is a very hard inverse problem that actually is mathematically intractable. MRI measures things in 3 dimensions. It's measuring things in the Fourier domain. So you actually get measurements of the brain activity that occurs in each one of these little tiny cubes throughout the brain. And these cubes here are actually the actual physical manifestations of what we're sampling using a brain viewer that my lab has published called PyCortex. Download and play with it if you want to for yourself. And the cool thing about measuring things in 3 dimensions is that it allows you to distinguish signals that occur at different points on the cortical sheet. So for example, in MRI, I can tell that this voxel here is recording from this point on this gyrus. And this voxel here, which is pretty near it, it's only 1 voxel away, is recording from this other point on this other side of the gyrus. And this voxel here is actually recording from a completely different gyrus, right? This gyrus and this gyrus are different, which is sulcus in between. Why is that important? To do with the wiring of the brain. So back in the mid-90s, David Van Essen had the brilliant idea that the folds in the brain actually are caused by the connections between the brain areas because neurons act like springs. So initially in development, you have just this ball of sort of protocortex, and nothing's really talking to anything else, but what happens is different areas of the cortex, they send out processes and try to connect to other areas of cortex. And so you'll end up with connections that are fairly strong and connections that are weaker. And the strong connections, because every connection is essentially trying to make itself as short as possible, it's essentially like acting like a spring, because it's trying to minimize its wiring length. The strong connections tend to cause gyri, and the weak connections cause sulci. Right? So here you see there's a really strong connection between these 2 brain areas. So they're on opposite sides of a gyrus. This is a gyrus. And there's a very weak connection between these 2 areas. They're on opposite sides of the sulcus. So what this tells you is in 3D, if you look at the brain in 3D, if you have 2 brain areas that are on opposite sides of a sulcus, they probably don't have a lot to do with each other because they have a very weak spring. They're not pulling very hard. If you have 2 brain areas that are on opposite sides of a gyrus, they have a really short connection to each other, so they probably have a really strong connection, and they're probably doing a very similar sort of thing. So if you're going to try to image the brain, you need to know, you really want to know where information is distributed across the cortical surface because you want to discover these maps. And you want to be able to distinguish things on opposite sides of a sulcus from opposite sides of a gyrus. And MRI allows you to do that because it's measuring things in 3D. That's why measuring things in these different locations across the gyrus and sulci are really, really important. Okay? Is that all clear? Plausible? Or you think it's kind of BS? Yes?
So, you mentioned earlier today that it's actually difficult to tell where the blood flow, or well, I should say, what region of the brain is causing the increased blood flow in this area. So,
well, it's kind of what I said. Okay. What I said was sometimes you can have the neurons that are influencing the blood flow be distant from the blood flow, meaning not in exactly the same voxel.
Okay.
And that's because the voxels are a couple millimeters across, and most of your modelable signal that you can explain with a theory or a model, it's not random, it's not noise, is going to occur in blood vessels about a couple millimeters across. And the capillaries are actually columnarly organized, and they're much smaller than a couple millimeters. So what happens is you have, if you think about it, I have my big thing, and it's measuring from a bunch of capillaries, say 3, and these are measuring from little tiny capillaries. And the columns are going to be organized according to capillaries here. But I'm pooling information to get where I actually measure. So it could be that I'm getting signal from an area larger than my voxel size. Right? It's just something to keep in mind. It's not really going to matter for this very much because there's nothing you can do about it, right? And in science, if you have some measurement device that's the best measurement device you can build, but it sucks in some respects, then you just have to live with it.
Okay, but so reliably increased blood flow in some voxel correlates with increased activity in that voxel, is what you're saying?
Reliably increased blood flow in a voxel is correlated with increased activity of the mitochondria in some area nearby that's feeding into it.
Okay.
Right? And remember, ATP gets made in mitochondria, and mitochondria are not just in the cell body, they're also in the synapses. So MRI actually measures, you can have a synapse that's in a completely different area from the cell body. So MRI can measure kind of different signals than you would measure as a physiologist. That's kind of a problem. And none of this is, you just got to keep it in mind when you're building a model, right? Okay, so just to show you that this method is not random, this is a study done by Roger Tutel and Russ Leiboway in 1988, and this shows a map of space on the surface of the macaque primary visual cortex. They used this stimulus. This is a little checkerboard line drawing, the checkerboard flipped on and off, and they injected the animal with a radioactive tracer bound to sugar, and as the brain processed this visual stimulus, the neurons that were active selectively bound the radioactive tracer, and then they just developed the cortex as if it was a piece of film, and they ended up with this. You can see that there's a topographic map of the visual cortex on the surface of the cortex. Now, only 7 years later, Marty Serino and Brian Lindell and a bunch of other people did retinotopic mapping. This is a retinotopic map. Here's the same angle map you see here, an angle map. And you can see that there's a map of space on human cortex. And they did this completely non-invasively, with no radioactivity or no development or anything, just in the magnet in a human. In only 7 years, we went from this, which really seems like primitive technology, something you get from the 19th century, to this, which really seems so the MRI is a really useful method. It's got its limitations, but you can do a lot with it. So there have been really 4 periods of MRI so far. There's been this period of functional localizers. And those of you, I don't know, does anybody here know about MRI or used it? Almost nobody. Yeah, this school doesn't have a big MRI community. So functional localizers are you might want to find face areas, so you would show places, pictures of places, and you show pictures of faces. You calculate the brain activity of both of these, you subtract them, and then you just find what responds more to faces. That's a functional localizer. NBPA classifier is essentially a functional localizer that's a little more sensitive. A bunch of people now do resting state correlations, which is you put something in the magnet, you just record brain activity, you don't give them a task. You look at the covariance of the brain to try to understand what the brain's doing. I wouldn't recommend that. There's also, you can try to look at the correlations of cross subjects. It says cross subject correlations to try to find brain areas. All of these methods have been used. They're all kind of insensitive. So the method that my lab uses is what I think of as a more sensitive method. And I'll explain that by showing you an example of the data. Let's go back to the data and figure out what we're recording. So on the lower left here is a movie. People are watching this movie while they're fixing on a small green spot in the middle of the display, and they're not asked to do any task here. And on the right is this brain, human brain. Now the brain, remember, is inconveniently folded up inside the skull. So since we have this, we have computers, we can basically take the brain data and we can inflate the brain, then we can put some cuts in it and we can flatten it out. So now we have the 2 hemispheres of the brain. Red here means areas where we have more brain activity, more blood flow, and blood oxygen use, and blue means less brain activity. And we can zoom in here. All the little polygons you see here are the individual voxels. They're now distorted because we've gone from a square to a distorted, flattened product here. And now in white here, we can impose the various brain areas that are known from retinotopic mapping or functional advisors and so on. This is the visual system in the back. These are the retinotopically organized visual areas. These are the higher order visual areas. This is the auditory cortex. This is somatosensory and motor cortex. This is some frontal eye fields and Broca's area and some other stuff around here. This is prefrontal cortex, which is involved in abstract thought and planning. Now you can just look at this movie and look at the blood flow, and the question's pretty straightforward. Okay, these are my x variables, these are my y variables. What's the relation between x and y? Right? This is just a big, ugly, hideously complicated multiple regression problem. Now the trouble is, you don't know what in this movie is driving blood flow back here, right? So here you see some flames, and now prefrontal cortex is active. Well, why is this active? Is prefrontal cortex afraid of the fire? Does the person realize, oh, wow, that was a movie of the Hiroshima shrine, and that's really horrible, and they start thinking about nuclear war? It could be almost anything. You have no direct way to know. Yes?
So you're saying here some areas have more activity, some have less. What's the standard compared to what?
There is no compared to what? It's just relative over time, right? As this movie is playing, this is changing. That's all you know.
So is this the change, or is this, when you say more than what?
This is all the individual voxels are z-scored for the whole time series. And this is a standard thing that everybody does in MRI for a reason. It's not a good reason, but it's a reason. Okay, MRI is measuring changes in blood flow, right? Well, if you just think about it, what do you think is going to cause the biggest change in blood flow in your brain? Of anything.
Pulse.
What?
Pulse or something. I don't know.
Pulse. Actually bigger than pulse. Changes in blood pressure. It turns out that you have, that your blood pressure changes with a time constant of about a minute. Even while you're just sitting here, it changes hugely over the course of about a minute. And nobody knows why, and nobody knows what controls it. I call this the God variable. It's just there, just floating around randomly. So your brain's like this fixed skull, and you're shoving more blood in it or less blood in it. So the bold signal is actually changing with blood pressure. It's also changing with pulse. It's also changing with breathing. All these things influence these signals. You don't care about these things, and you don't really know what's causing it. So what does everybody do? This in MRI, in math, this is called a blind source separation problem. I have signal that I care about, which is the component of the bold signal that's related to neurons. I have signals I don't care about, which is everything else. And I don't know how to measure those things I don't care about. So what do? Well, what everybody does is they just say, screw it, I'm just going to z-score all my variables, and the stuff that survives that is the stuff I care about, by definition. That's what everybody does. Every MRI package does this as the first. You also need to detrend. So a lot of people will linearly detrend over time, but of course your blood flow is changing as a random variable. If you're in the magnet for an hour, linear detrending isn't the best thing, so then you might say, well, I'm going to detrend with, there's a million things you can do. Some of them are better than others, right? Is that clear? So these are just z-scored. All right, so that's why average is 0, right, in this metric. And the question, yes?
So if the same patient is watching a video and that fMRI is working, is there some sort of reproducibility?
We'll get to that, but that's the critical question here, right? Is there some sort of reproducibility? And if there wasn't, then there would be nothing to talk about, right? So if you think about this random variable that's blood pressure, that's presumably not reproducible, because that's just a random variable. And then if you think about the causal part of this, that should be reproducible.
Because you can think that each brain would be unique, right? Like each individual.
Oh, so you're talking about reproducible across people?
No, of the same person, of the same exact person.
So, so here's the way to think about it. In psychology, there's this idea of states and traits, right? And a trait is like your stable personality characteristics, and a state is the manifestation of your stable personality characteristics in this particular environment because stuff's happening, right? And the same goes for MRI. You have stable components, and then you have these non-stationary components over time. So if you can imagine if the person, the first time they see this movie, they've never seen it before, their brain might actually respond differently than the 5th time they see the movie. That would be a non-stationarity and would not be reproducible. So some parts are going to be reproducible and some parts aren't. Now, I'll explain how I deal with that. How science deals with that, how psychology deals with that is actually pretty bad, but there's a better way you can deal with it that I'll get to in just a second. Okay, so just hold that thought for about 3 slides. Okay, so now we've got this multiple regression problem, right? We have a bunch of pixels on our screen, and we have 60,000, say, cortical voxels that we measure. And we want to find the relationship between the stuff on the screen and the voxels. But It may not be that you can describe each voxel in terms of the pixels. You may need to make some more abstract representation. Like maybe you're thinking about, what the person on the screen, whether they're happy or sad. You're not going to extract that from the pixels. You have to infer that, so that's going to be a higher order variable. So how do you do this? Well, this is multiple regression. You're allowed to essentially solve this problem any way you want. Some ways are good, some ways are bad, depending on your data. Regression is one of the oldest areas of statistics. But this is a very simple way to think about it that makes it easy to kind of get your brain around this problem. Here we have a stimulus space. This is just a bunch of pixels, right? So there's a bunch of pixels, and each image is going to be a vector in this space. We have a brain. Of course, we didn't measure the brain. We measured some brain activity, which is a reduced version of the brain. So this is going to be the activity in voxel 1, the activity in voxel 2. And so each pattern of brain activity is going to be a vector in this 60,000 dimensional activity space. This might be a 1,000,000 dimensional pixel space, 60,000 dimensional activity space. We want to find the relationship between stimulus space and activity space. And the only thing we know is this is nonlinear. Because if this was linear, there would be no point. You could just do it with one neuron, right? Linearities are stupid. They're information preserving. You want your brain to extract information. It's going to be something nonlinear. So if you want to determine what this nonlinearity is, one thing you can do is you can propose, as a theory, you can propose a feature space. You can say, well, I bet that area X has this particular kind of features. Like maybe it's a face detector. So I'm going to run a nonlinear face detector over my images and go from a pixel space to a face space. And that would be a feature space. And this is going to be a nonlinear transformation. And then you can use regression. This has nothing to do with your data. But then you can use regression to try to map the feature space onto the activity space. And I would argue, I would submit to you, that what you want to do is find a feature space such that the relationship between the feature space and the activity space is linear. In other words, this is just a simple linear transformation. And the reason for this is that this tells you what information is explicitly represented in an area. Remember, a linear transformation, like just adding 10 to all the numbers, has no effect on the amount of information in your signal. A nonlinear transformation can add information or it can remove information. A linear transformation is information preserving. So if you find a feature space that you can predict the activity of it perfectly linearly, then you've basically found all the information that can be extracted from this brain area. And this tells you also what the brain area does. It does this particular feature transformation. So you can think of the nonlinear transformation from the stimulus to a feature space as your hypothesis. You can think of the activity space as your experimental measurement, and this is just linear regression. Now, if you do find a linear encoding model which goes from features to activity, as I'll show in the end, you can always do decoding from the activity space back to the feature space. This is completely symmetric at this point because it's linear. Is that kind of clear? Right. It'll be clear when I give you an example. So for tonight, I get bored. I get bored really easy, and I always want to talk about new stuff. So I'm talking about unpublished stuff. And the unpublished stuff is how you use this method to try to understand how people comprehend narrative speech. And the stimulus in this experiment was really simple. It was these moth stories. If you don't listen to the Moth Radio Hour on NPR, you should. These are awesome. The Moth is like stand-up storytelling. That is, people get up on stage and they tell these stories. They're usually autobiographical stories about things that happened to them. Some of them are funny, some of them are horrible. They're about redemption and heroism and family conflict. They're really compelling stories. And all that happens in this experiment is people go in the magnet and they just listen to these stories. They listen to like 2 or 3 hours of these stories over several MRI sessions. They don't have to do anything. Subjects love this experiment because they can just lie there and listen to the stories, and they're really great stories. They don't have to fixate, they don't have to do a task, they just listen to the stories. Now, one of the stories we actually repeat several times throughout the experiment. So we take all of the data that's not repeated, and we're going to use that to fit our model. And then we're going to take the data that's repeated, and we're going to use that to get predictions, okay, and to evaluate predictions in a framework that I will explain here. So you can see most of this. Okay, so we have 2 data sets. We have an estimation data set. If you're a computer science person, it's called the fit data set. We're going to use this to estimate a model for each and every voxel in the brain. And then we have a validation data set. If you're a computer scientist person, you know this as the test set. And we're going to use this to test the predictions of the model. Okay? So how do we do this? Well, the stimulus here is the stories. Now is the time, blah, 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 whatever the story was. And so let's ignore the brain data for a second. We take the story and we project it into feature spaces. And these feature spaces are hypotheses about how the brain might represent this story. So here's a feature space that's the spectral, the spatiotemporal spectrum of the feature. So this is going to be the short time Fourier transform. This is essentially the way your cochlea represents these stories. It's representing the short time Fourier transform of the amplitude of the waveform. Right? And there are about 500 spectral features that we're going to transform the stimulus into. And then you figure, well, the stories, obviously you can't tell much from the story just from the sound spectrum. You must extract higher information from the story. So probably you subtract phonemes. Somewhere in your brain you must have phonemes being represented. So we can take the story and project into a phoneme space, and there will be about, well, there's 57 phonemes in English. Okay, so we have a 57 phoneme space. Now we take the stories and project them into syntax. I should tell you up front that our syntax model that we have is really bad right now. It's a hidden Markov model for those of you who know about linguistics, and it doesn't work very well. So when I talk about syntax, you should remember evidence is not evidence of absence. We have not tried very hard to make our syntax model work, and as you'll see, it fails miserably. But that's because we haven't worked on this very hard. And then we have a semantic model. Semantics is the meaning of the stories. That's what semantic means, meaning. And in this particular case, we have 985 semantic features. How many people know about linguistics? Oh, shoot, enough people that you actually probably don't know how we do this. So there's a corpus of English called Simple English. It's the 985 most common words in the English language. And we essentially use those 985 words to do something that is roughly comparable to something you linguists and computer scientists have heard of called latent semantic analysis. And this essentially just finds the covariance between these common English words. And we do this inside a corpus of 6 billion words. So we're essentially modeling the meaning of each word in terms of its covariance with these other words. Okay? How we exactly do this doesn't really matter. Basically, we have 985 semantic dimensions, okay, that talk about the meaning of English. Now, this adds up, this looks like maybe about 1,600 or so dimensions, right? And what we're going to do is, and we've done nothing with the brain data yet. This is just transformations from the stimulus into these feature spaces. And now we're going to take each individual feature space, and we're going to take the data from one voxel, and I'm going to shove it down here, and we're going to find a weight that tells us how much that individual feature affects the bold response. So for every one of these 1,600 features, we're going to basically build a little function, a little hemodynamic response function that says, when this feature appears in the story, how much does the brain activity change at this specific location in the brain? And we do that for all 60,000 voxels in the brain. So the cool thing about this is it'll tell us, the voxel will tell us what features it cares about. We don't have to assume anything about the features. We just say, here, you can pick any of these features, these 1,600 features, you tell me what features you care about. Okay. So now you fit the model. This is just regression. And now you get signals in red here, okay, that are the predicted signals. Now I take a new set of data with different stories, and I shove these stimuli through the model, and I get some predicted responses. And I look to see how my observed responses compare to the predicted responses. And this tells us which features are actually important for the voxel. Right? We're trying to essentially predict variance in the voxel using this cross-validated data set. Remember, for those of you in psychology or biology who haven't taken a lot of statistics, statistical significance is kind of what everybody usually tries to go for when they're publishing a paper, but they shouldn't, because statistical significance is not significant. Statistical significance just tells you your data is not random. So whenever you hear somebody say, my data is statistically significant, inside your brain you should convert it to, my data is probably not completely random crap. That's what they're really telling you. That's not what you want to do. That's a low bar, right? The reason everybody uses it is because it's easy. What you want is a prediction in a cross-validated data set. What you want to say is, okay, I have a model, and I get some brand new data, and all of the potentially explainable variance in this data I can explain with my model, right? So you want to collect some cross-validated data. You want to calculate the noise ceiling, because since it's a finite data set, you'll never be able to predict it perfectly. So maybe the noise ceiling is like 90%. You can calculate this if you have repeats. So you can say, of the data I have, the perfect model should be able to predict 90% of the variance in this data. Now, I have a model. It predicts 70% of the variance in this data. I know exactly how happy I should be. I should be 7/9 happy. You get to 9/9, you're done. Okay? So when you're doing predictions, you always know exactly how happy you are. When you're doing significance, you just know you're not random. So don't be those guys. Don't be satisfied with significance. Significance is necessary, but not sufficient for doing science. All right. So now you can take all of these models. Each voxel now has, well, here I only showed you 3 models, the phoneme model, the syntax model, and the semantics model. And each voxel has predictions of its bold response from these 3 models. And so now we can code the phoneme model in green, the syntax model in blue, and the narrative model in red, where each of the RGB guns is essentially telling us how much of the variance in that, that feature space predicts in this cross-validated data set. And then we can plot on the flat maps here. So remember, these are 2 hemispheres. This is the visual system here at the back. This is the auditory cortex using a really generous visualizer, so it's a bunch of auditory areas. This is the motor semantic sensory strip. You can see that the phoneme model predicts really well right here in the middle of this auditory cortex. And this turns out to be A1 and A2, primary auditory cortex, secondary auditory cortex. Exactly where you would expect, based on everything we know, where you would expect phonemes to be processed. The syntax model doesn't predict well anywhere, but somewhere in your brain there's probably syntax is represented, right? We just haven't worked very hard in this model, so we don't know where it is. But this narrative semantics model, this 985-dimensional feature space for semantics, you notice, fits really, really well in a lot of places in the brain. This is down here, sort of posterior auditory cortex between auditory and visual cortex. This is the temporal parietal junction. This is parietal cortex. This is over down the midline, the retrosplenial cortex and the precuneus. So this is basically a strip down from here all the way up and down into the midline. And then all this stuff up here is prefrontal cortex. So this semantics model seems to be really good and very general. So the graduate student who was doing this project, Alex Huth, decided, oh, I'm just going to pay attention to semantics and just ignore all the other models. So let's look at the semantic model. The first thing you can do is you can take an individual voxel and you can visualize the words that the voxel likes versus the words that the voxel doesn't like. So here is the voxel. It's located at the end of this arrow. This is actually probably in something, I suspect, a Broca's area or something nearby Broca's area. And these are the words that are predicted in this cross-validated set that the voxel likes, where size and saturation or contrast tell you basically how much this word is predicted to affect the bold response. You can see that this voxel is suppressed by politics, political, religious, and appreciated in opinion, just like politics. But it really likes maximum miles, 8 upwards, nearby meters. It seems this voxel seems to be tuned for numbers and distances maybe, and it doesn't like politics. Now, okay, so we can go through the brain. There's only 60,000 of these voxels. That would take till next year, right? So we don't want to do that. So we're gonna do something with this data. We got 7 brains. Each brain has something like 50 to 80,000 voxels. This is too much data. You gotta do something with it. So in science, when you're faced with this problem, you always think, I gotta do dimensionality reduction. I have to take these data sets and reduce them down to some set of dimensions that I can think about. And since all the data here are Gaussian, remember I said we z-scored everything, so it's all Gaussian. The dimensionality reduction you always do first when you have Gaussian data is called principal components analysis. And principal components analysis, some of you have heard about, it finds a set of orthogonal dimensions, they're independent orthogonal dimensions that account for the variance in the data from the dimension that accounts for the most variance down to the dimension that accounts for the least variance. And when you do this across these data, you find out that you can reduce all the data for each individual subject down to about 100 semantic dimensions, 100 principal components, excuse me. And that accounts for about 95% of the data. Now, each person's principal components are just this vector space, right? And so I can take the vector space from person 1, I can take the vector space from person 2, and I can look to see how they line up. And when you do this, you find out that all subjects share, where all subjects means the 7 graduate students in my lab that ran this project. So we'll assume that's all humans for the purpose of this talk. All subjects share the first 5 or 6 principal components. They're almost exactly aligned. And after that, the principal components are different between individuals, probably because of noise and because of individual differences. So Alex decided to focus on just this first set of 5 or 6 principal components, because that seemed to be the common semantic representation that was similar across all different humans. Okay. So when you do this, now these principal components are a vector space that allows you to visualize the data. Because you can take, say, a couple principal components, and you can take the tuning curves of the individual voxels, or excuse me, you can take the words, and you can project them into the principal components, or you can take the voxels and project them into the principal components. Either way, this space mediates between the features, the 985 features, and the voxels. So let's first look at the words, the 985 features predicting the principal components. And this is just as an example. Here's PC1 and PC2, and he's actually coded the words according to their loading on principal component 1, 2, and 3 RGB coordinates. So you can see if you kind of march around here that the words differ. So here we have visiting, arrived, meeting, waited, apartments, talked, parents, husband, father, confessed, arrested, daughter, married, refused, murder, child, convicted, murder, innocent, victim, cruel, kill, repeatedly, accidentally, male, protect, human, vile. Then we get into a real change. We get American, explode, fielder, poison, lethal, expressive, tongue, subtle, breast, glove, skin, blade, thin, metallic, colored, layer, smooth, edges, tops, wide, intricate, jackets, lighting, clouds, atmosphere, landscape, floors, winter, garden, shop, scenery, spacious, summer, facilities, then rumbling, exploring, dreaming, explore, moments, reading, experiences, awake, studying, quiet. So this space, remember, comes from the brain data. It doesn't have anything to do with the features. We've taken the features.
Just a time check, we have only roughly about maybe 5 more minutes or so.
Well, we're going to get through part of it. So how about 10 minutes? You guys can leave whenever you want, by the way. Okay?
Yeah.
So really, feel free. So this data comes from the brain, not from the model. You can also project this onto the surface of cortex. So here you see there are red areas that are responsive to families and family drama, and green areas that are responsive to texture and object, texture and structural kinds of information. But now we have a problem, which is we have 5 brains, each of which have this common space, but if you notice, the patterns are different across the brains, right? So now the question is, how do we make the data common across the brains? So this is a classic problem. We want to know where are the areas across these people, and how can we compare the results? This is a classic problem in MRI, and it's known that this is a problem because brain areas differ. So this is an old study from Brian Weindel's lab that shows primary visual cortex across 3 individual subjects. You can see that primary visual cortex varies somewhere between 250 and 400% across people. So how do you morph brains into each other if you have all these very different brain area sizes? This is a problem. So the standard way you do this is you do something called, it goes by various names, brain morphing or volumetric alignment. So in color here is the average cortical folding of 1,000 people if you just average them together. And in gray here is one individual brain. You can see that this folded average brain does a poor job of representing this one individual brain. You can see this brain has sulci that are much deeper than the average brain. And that means that you can't just average the brain, you can't map the individual brain into the average brain without losing a lot of information. So we came up with this method of doing this that uses something called a generative model. This is almost done. The idea here is, well, imagine that the brain is like a pizza with a bunch of pepperonis on it, and the pepperonis are just areas that are homogeneously tuned for different kinds of semantic concepts. So now I can represent each pepperoni by its center location, and I can imagine that if I have one brain with a bunch of center locations, I can connect these together with springs, and then maybe I can take this spring and ball arrangement and apply it to another brain and allow the springs to relax in such a way that the map will be, essentially the areas will be relatively the same across brains, but they will morph around depending on the specific brain anatomy. And this is just a physics problem called a Boltzmann machine. So it's pretty easy to implement using a generative model. So here we have 5 subjects. What happens is you lay the areas down randomly in subject 1, and then you have a bunch of springs connecting the areas, and in subject 2, 3, and 4, you just try to make in the initial setting, you just try to make them as similar as possible. And then this thing settles down, and it settles down so that the likelihood of these individual areas is as high as possible given the functional data that you have. And it settles down across brains here. So this is not a discriminative model. We're not averaging brains together here and taking the average brain and the variance. This is a generative model. We're generating the underlying probability distribution that emits individual brains as samples from the generative process that generates brain areas. Got it? So very different kinds of statistics. The cool thing about this model is it allows us to predict where the areas are going to be in a completely different brain that we didn't use to build this model. So we have 4 brains here that we used to build the model. Now we can take a 5th brain that the model never saw, and we can give it to the generative model, and it will splurt out the areas where it thinks they should be. And then we can compare it to the actual functional data. So here's what you get for 3 subjects. Here's the functional data on the left. Again, these are RGB projections of each individual voxel into this common principal component space for the first 3 principal components. And here are the brain areas that the model assumes should exist using exactly the same color scheme. And in each case, what happens is this subject was held out. So here, we generated the models with subject 1 held out. Here, we generated with 2 held out. Here, we generated 3 held out. So there's no possible overfitting going on here. And these are the likelihoods. Red here means that the model is very likely at this location given the data, and blue means the model is very unlikely given the data. You can see that the model actually fits really well in these areas exactly where we think it should, in prefrontal cortex and in this temporal parietal strip. So this model's great. It's just about the last slide. Oops, I don't know why that's there. It's out of place. So now let's assume that our model's correct, and just take all 5 subjects together, and because we don't have that many subjects, throw them into the model and generate the brain areas, and we get something like this. So here we've removed the areas from regions of cortex that were not statistically significant. This is an added step that has to occur at the end. And what we end up with here is 113 distinct functional areas tiled around prefrontal cortex and this strip from temporal cortex, temporal parietal junction to precuneus. In the left hemisphere, and 123 functional areas in the right hemisphere. These are distinct functional areas, and they all have their own individual tuning as given by this color code. The color is the projection of this principal component. Now, of course, you want to know, well, great, I just found out 250 new brain areas. What do they do? Right? That's kind of the problem. This is a problem that everybody who does neuroscience has. Like, you record from a single neuron. I record from a neuron in V4. To build a good model of a V4 neuron, say 60-70% of the variance, I might need a model that has 1,000 parameters, and now I have to explain to you what this V4 neuron does. It's really difficult, right? So the same problem applies here. I've got these areas. They all are tuned for a 985-dimensional feature space, and I have to tell you what that feature tuning is. So one of the shortcuts way we can do this is we can say, well, I can use these tuning curves to predict what word would be most likely to generate the largest possible activity from this brain. This is like finding the peak of the tuning curve. And we can kind of plot that on the cortex for different brain areas. So here you see this is I'm zooming in on the left hemisphere here. You can see that this little blue area, these are the words that it predicts to activate the most: atmospheric, scenery, clouds, breeze, and stream. Thinner, thick, thicker, lighter. All of these, for different distances. Here's a murder, victim, innocent, murder, die area right here. I'm not sure if I have it on the next slide. Let's see, did I put that in here? Oh, right. I actually had to add this because Alex didn't do it. There's an area down here, left prefrontal cortex 11, that like sin, Jesus Christ sins and prays. And right next to it, the left prefrontal cortex 10, and there exists scientific. I should mention, if you notice, that this 11 is much larger than 10, so that's so that's the left hemisphere. In the right hemisphere, you see there's more sort of social drama in the right hemisphere: wife, husband, arrested, murdered, on husband, wife, pregnant, reduced, wives. But there's also a lot of time, 10, and number, 10, 5, 26, 3, date, hours, month, hours, murder. So, okay, so that's that. So you can go through this forever, right? Now you might ask, well, wait, we just reinvented phrenology, and we did that 100 years ago, and it sucked the first time. Why are you redoing this? The thing to remember is that phrenologists had the idea of localization was a reasonable idea. They didn't come up with that. That was from neurologists. Neurology started in the 19th century when muzzle velocity of firearms got high enough that a bullet could go straight through you without killing you, and then that provided a bunch of natural experiments for the neurologists to go in and find what different lesions of brain areas did. And that was the beginning of the idea of localization. And the phrenologists, what they did is they took the idea of localization and they just did a really crappy job of solving it, right? But with MRI, we can actually solve localization in a much better way. The problem is that we're limited by this feature space. We've only got this 985-dimensional feature space. And all of our interpretation of these areas is a projection of whatever that area really does into this feature space. So if I hear about a story that has to do with murder, and my thought is, oh wow, that poor guy, what's going to happen? He's going to go to jail, and you know, he's going to be in jail for 20 years, and this is so sad. And I start having thoughts about that every time I hear about murder. In this experiment, I'm going to think that that voxel cares about murder, even though that voxel might actually care about thinking about the consequences of being away from the person's family for 4 years, right? So we don't really have full knowledge of what the features are until we get features that predict 100% of the variance. We're always stuck in these feature space representations. Also, you might note that these don't look like hard edges. They look like gradients. In fact, we published a paper last December saying that areas were a dumb idea, that everything's gradients. And I just showed you an area model. So you might say that I'm not being consistent. But the point is, you could take this area model and you could turn it into a gradient model, and you could fit gradients instead of areas. And the last thing that I'll mention in this talk is that these areas are not necessarily stable. So this is from a paper we published last year. This is a semantic map that we got from movies. So we had people watch movies, and we fit a 1785 dimensional feature space based on this hierarchical inheritance tree from linguistics called WordNet. So now each individual voxel has a tuning curve in a 1785 dimensional space. You can take the first three principal components across individuals. They're shared. Well, actually, principal components 2 through 5. And you can color code them. And this just shows you what the semantic coding of the different areas is in vision. Red here happens to be motion. And green is people, and yellow is animals, and blue is buildings. Now you can have people watch movies, and you can tell them, okay, when you're watching this movie, whenever you see a building, hit the button. Or whenever you see a person, hit the button. Whenever you see a vehicle, hit the button in 2 different conditions. Now you take all the data and you run it through this, this framework, and you can actually get the new tuning under these 2 attention conditions. And here you see that when your target is humans, all of cortex becomes much more tuned to humans. And when your target is vehicles, all of cortex becomes much more tuned to vehicles. So what's happening is the tuning curves of these voxels are shifting depending on the state of attention. And that actually has to happen given, I mean, for reasons I can go into if you want to know any questions. So that's about it. Different individuals share a low-dimensional space. These neurosemantic features are represented broadly across cortex. We have about 250 distinct functional areas just for this one task. We don't know how stable the maps are. We don't know how the interpretation will change with other features. And we don't know how the interpretation will change when we use a different gradient-based generative model. And that's about it. If you want to know about decoding, I can talk about that, but that's you guys should vote, or people should have a chance to leave if you don't care. So I'll just stand here for a few minutes. Okay. So if you want, well, you can applaud. So maybe wants to take off. The decoding part takes about 5 minutes. It's entirely up to you. I will not. I don't know anybody's name, and I can't grade you. So really,
what's the gist of this?
The gist of this is you can do decoding.
By which you mean going from your feature space to putting things onto the brain, or?
No, it means decoding what you saw, right?
Oh,
what
okay.
you experienced.
Okay.
This part is 5 minutes, I would say.
So really, if you can stay or go, you know, it's up to you.
It's kind of like taking all of the science and using it for something that some people might consider like practical, some people might consider kind of useless, but it's cool looking.
So I'll just talk while people eat. That way there'll be less offense. Okay, so what we had before is we had a stimulus, and it generated some brain responses. And we built an encoding model. The encoding model goes from stimulus to brain responses. So we have a stimulus, we nominally map into a feature space, and now we take those features, we linearly regress them onto the bold responses that we measured, right? That was science. If we do this, and we can perfectly predict bold responses from the feature transformation of the stimulus, then we're done with science, right? Science is, we have an explanation, which is our model, we have predictions. But once you have this encoding model, you can actually do decoding. The way you do that is you take all your bold responses that you measured, you weight them by their likelihoods, you linearly map them back into the feature space, and now you can just look at the features. Or, if this nonlinearity is invertible, you can actually go from the feature space back to the stimulus space. And why can you do this? You can do this because of Bayes' theorem. So Bayes' theorem, which you can't see at the bottom here, tells you that if you have a likelihood, which is your model, gives you a probability of the response given some function of the stimulus, you can multiply it times a prior, you can get a posterior. And posterior gives you a function of the stimulus as a function of the response. So you can always do this. Whenever you have an encoding model, you can always build a decoding model, right? That's the point of this. These two things are not dissociable. So here's an example of encoding. This is an encoding model that uses a model, it's a hybrid model. It uses a model of low-level vision to get the edges of the stimuli, and it uses a model of higher-level vision to get the semantic category of the stimuli. On the top here are 3 images that we showed people, and on the bottom are our reconstructions. This is like a harbor scene. This is a harbor scene with reverse contrast. There's a bunch of people in a room, with their hands up. 2 buildings in the front, building in the back. 2 buildings in the front. One happens to be the Coliseum, building in the back. So these reconstructions are pretty good. They all look kind of like buildings, but they don't look like the buildings that we showed people. So to explain why that happens, it takes a few seconds. Remember, I said to do decoding, you need to take the models, and you need to multiply them times a prior. So if we want to decode images, we need a prior on natural images. Turns out no one knows what the prior is on natural images. That's actually the goal of computer vision, but they haven't solved that yet. So we don't have a prior on natural images. So what do? Well, what we can do is we can just take a big gigantic library of natural images and treat that as our prior. So what will happen is we'll troll through this library of images, and we'll find the image which, based on the brain activity, we think is most similar to the image you actually saw. But these images are not in that prior. So we can't actually reconstruct these images. So we have a library of 50 million images we got from our friends at Flickr. Just gave it to us on a hard disk, so these might be your images actually if you put on there. And we just troll through and we find the images that we think are the most similar, and that's our reconstruction. Here's another decoder that actually uses high-level semantic brain areas. This uses a computer algorithm called latent Dirichlet allocation. You do not want to know how LDA works. It's way too confusing. But the point is, LDA tells you about scene categories, and it actually has built into it, it learns the statistical relationship between objects in scenes and scene categories. So we can now take a new image that the person never saw. Here's a guy with a tarantula on the side of his face. This was not in the images we used to build the model. We can measure the guy's brain activity, and we can decode, well, I think this is land animals, few people, and maybe food, I'm not sure. And we can decode the objects, man, woman, person, head, animal. You can see in all these cases, the decoder works really well. Here's a sporting event, large crowds, few people, electoral, athlete, people, man, woman, person. This all works pretty well. This is only decoding information from high-level brain areas. Here's a motion decoding algorithm. So this decodes information only from primary visual cortex. So here we have the images we showed. Is up here on the left. It's the presented thing. This thing down here on the right is what you would do if you used a much more primitive and simple brain decoding algorithm called direct decoding. And here are our 2 decoded results. This up here is called the map, or the maximum of the posterior. In this case, our prior was 5,000 hours of video that we downloaded from YouTube at random. So these images were not in that 5,000 hours of video, so we're trolling through this 5,000 hours of video to find the piece of video that we think is as similar as possible to what you saw. And on YouTube, there's a lot of people, so if this is a people, then we decode it really well. But in our 5,000 hours of video, there's no elephant, so it can't do a good job decoding that. So in that case, what we do is we take the top 100 clips and we average them together and we get that. So either one of these 2 things are good decoding results. And the neat thing about this is, remember I told you that this bold activity is really slow, right? It takes a couple seconds to appear. So the fact that you can decode video seems like kind of a miracle. It seems like you should be able to do that. But natural images, natural movies have very high spatiotemporal correlations. So even if we're only sampling the brain every second or so, we can infer what happened during that intervening time. This is a semantic decoder. Here we're decoding 1,785 nouns and verbs that appeared in the video. We're decoding them simultaneously, so I'm just laying them out on the first 2 principal components here. So you can see this works pretty well. Talk, we get man, hand. Okay, it's not perfect. Mountain. You might say dog. Wait, there's no dog. What the heck is wrong with that? But if you see there's a dog. What happened is we overcorrected for this hemodynamic delay. And so it looks like your brain is actually anticipating what's happening in the movement. It's not really true. So here we have an ocean scene, we get body of water, cloud, vegetation. Here we're in Waikiki, we get building, road, mountain walk, people. This thing works pretty well. In fact, this thing kind of worked. This is where I started to get really scared, because I realized if there is something in your active conscious thought going on right now, we can probably decode it. And that's kind of a scary prospect. So this is imagery. I get asked a lot, can you decode imagery? So My postdoc, Thomas Nazarelis, spent a week just memorizing these 4 pictures. We just sit at his desk staring at the Mona Lisa, trying to make the perfect image of the Mona Lisa. And we put him in the magnet, and we just showed him the name Mona Lisa, and he had to try to make an image of the Mona Lisa. And then we're going to try to reconstruct the Mona Lisa. Now this is an interesting problem because the model we're going to use for reconstruction is a model that we built when he was actually looking at actual real images, although not the Mona Lisa. But when you're doing imagery, your brain must be doing something different than when you're doing visual perception. Because if your brain did exactly the same thing, you wouldn't be able to tell the difference between imagination and the real world, and that would not increase your survival rate. You'd be dead. You'd be eaten by a tiger. So we're using the wrong model, right? And the question is, how well do we do? So when we do reconstructions here, we get Salma Hayek from Mona Lisa, jockey for the soldiers, and doggy or for the cat, or whatever, vegetable fish. So this thing works. It works about a third as well, one-third as well as if we were decoding actual images the person saw. But still, that's pretty good considering we had the wrong model. Yeah.
Do you see certain areas as more important for imagery versus actual vision? So does V1 just shut off? Or?
So imagery seems, there was a lot of prior suggestion about this, and our data confirms that imagery seems to be generated basically from the midbrain, like the hippocampus and pericampal regions, and it's essentially the data that's stored there that has to do with memory gets essentially fed back into the visual system, top down. And so you end up with really good imagery results in higher order visual areas. But those visual areas don't really have retinotopic maps. You don't need to represent retinotopy in some abstract area that's, that you're using to recognize whether you're my mom or not, right? Because retinotopy is kind of irrelevant for that task. So if you want to, it turns out that as you go farther and farther back into the visual system, these signals essentially get weaker and weaker and weaker. And if you want to reconstruct these things, these are actually reconstructed here from primary visual cortex. These are reconstructed from higher visual cortex. But we have another data set where we recorded at 70, and then we were actually reconstructing primary visual cortex. So the signals are there even in primary visual cortex in V1, but they're very, very weak. And you kind of know this is true intuitively, right? So if you close your eyes and you try to make a mental picture of your mom, well, you can kind of do it, but for most people, it's pretty vague and woolly. And that's because you're not storing that really fine, detailed information. So for brain decoding, basically, think of it this way. This is the first photograph that was ever taken. It's by Joseph Niepce in the south of France in 1825. And you can see that there's a building here, and a building, and a building, and probably a horizon here. This is a lousy photograph, but you can tell it's a photograph. You can tell what's in here, right? And now, 200 years later, in almost 2025, photography is so good that no one even wants to pay for it. Right? It's just free. Just give me the photograph out there, right? This is where we are with brain decoding now. Right now, we can decode something that looks like this. It's kind of woolly and fuzzy. It's kind of the right thing, but it looks horrible. But just imagine what's going to happen in 200 years. Because remember, everything that I said, there was a symmetry between encoding and decoding. So every single thing that every neuroscientist learns about how the brain works allows you to build a better brain decoder. You can't have brain encoding without brain decoding. These two things go together. So everything that every neuroscientist does builds a better brain decoder, and that means that as long as NIH keeps paying for neuroscience, brain decoding is going to improve. And right now, we're at this stage, but in 200 years, who knows, right? So I think basically eventually there's, you can imagine somebody that invents something like a radar gun, but it's a brain decoding radar gun. So I pointed at you and I realized you're thinking very dirty thoughts right now. Right, and that is not outside the realm of possibility, because really all it would take is a way to remotely measure very subtle brain signals and solve the 2D to 3D problem. But if I have a gun that's actually measuring in 3D, which might happen, then it's good. So somewhere down the road, brain decoding is going to increase. We don't know where it's going to go. We don't know how long it's going to take. We don't know what the technology is going to be. But that's kind of how technology works, right? It works by punctuated equilibrium. So what I always say is you should be worried about this now, because this is going to have really big privacy implications down the road. And Americans are really bad about guarding their privacy. Anyway, so if you want to go on my website, which is gallant.org, you can play with this interactive, there's a bunch of interactive brain viewers on there that allow you to manipulate the brain. You can take it and move it around and look at it in your browser. You can flatten it out and make it folded up. And then you can click on this. This is a tree, semantic tree for WordNet for people watching movies. You can click on individual concepts here, like, you can click on priests and look to see how priests are represented. You can click on Ferrari and see how Ferrari is represented across the surface of the brain. Or you can click on position on the brain and see how it's represented in the semantic space. There's a bunch of brain viewer tools on there you can play with. You probably don't care about this. NeuroTree. Does everybody know about NeuroTree? No. Okay, so the University of Chicago is underrepresented in NeuroTree. NeuroTree is a giant genealogy of neuroscience. So if you're working in a lab, you're a grad student or postdoc, or there's no PI, so maybe one, then you can make sure your entry's on NeuroTree. So like if you're working in a lab and you want to know who is my academic grandfather, you can go to NeuroTree and type in your advisor's name, and it'll tell you the whole lineage of your advisor. And also, if you want the data, all the data from my lab is always posted eventually publicly, and the website for this is crcns.org. That's Collaborative Research in Computational Neural Systems. This is funded by NSF. And if you go to CRCNS, there's tons of data sets on there from mice and cats and macaques and humans that you can use for your own purposes. That's all data that's been published publicly. So this is all standard public access data set. You can play with all that stuff. And that's about it. I'm sorry for taking so long. Oh, this is Alex. He did all the work on this semantic model, and I feel like I have to point him out specifically, and I'm done. So, on the show.